This chapter focuses on the final group of organisms that we study in microbiology, and this will be the acellular pathogens. So this includes the viruses. Viruses are classified as acellular. They are obligate intracellular parasites. They have to have a host cell in order to reproduce. They cannot reproduce on their own, which is why they're considered intracellular parasites. Viruses, there are literally thousands of different viruses, but there definitely is what we call host specificity, meaning the type of cell that a virus will infect is very specific. And we'll get into that a little bit later, but just like there are a lot of, say, plant viruses that they infect certain types of plants, and even not just any plant, but maybe just a tomato plant or just a potato plant or just a tobacco plant. But those usually, if they infect plants, they usually won't infect animals, and certainly not humans. When we look at viruses, they either have DNA or RNA as their nucleic acid, but never both. And we will look at, is it single-stranded or is it double-stranded? That will help us with the classification. So it has this nucleic acid, whichever kind it has. Lysate will not have both. And then that's going to be surrounded by a protein capsid that's going to help protect it. There may be an envelope, but not always, that's surrounding that capsid. They come in all different shapes and sizes. Viruses are extremely small, and that is something that I know as students trying to get a handle of the size differences. A fungus is huge to a microbiologist as compared to a virus. So a virus is going to be the smallest thing that we're looking at in terms of the microorganisms. Virus is very, very small. Fungus is huge. Bacteria are going to be in the middle. In order to see a virus, you're going to have to use an electron microscope. There's just no way you're going to see it with a compound light microscope. You have to use the electron. They're just too small. As you can see here, this gauge here is 50 nanometers. This is tobacco mosaic virus. So you can see they look like almost these long rods. So there are several of the virus particles here. And over here you can see the type of disease that it causes on the tobacco plant. All living organisms can have some type of a viral infection. For viruses, like I said, their host, it's very specific as to which host cells it can infect. Usually that host range is fairly narrow. A bacteriophage or a bacteriophage, depending on where you are in the country, because there's differences in pronunciation, that's okay. What that is a virus that infects a bacteria. We use these a lot for studying viruses to get a better handle on like how they reproduce, what are some of the requirements of them. But a bacteriophage will infect a bacteria. And usually it's very specific too as to which bacteria they can infect. Viruses may cause disease in the host. It could damage the host. Or it may be that it infects the host and it just goes dormant. And the host is actually asymptomatic. You have no idea that the virus is there. That can also be a problem because sometimes when it's dormant and it's not showing any signs of infection, which would be asymptomatic, they can still be contagious and pass the virus on to others, which is really tough because you don't know that you have it. This is another electron micrograph. It's at 50 nanometers. This is a bacteriophage that is infecting a bacteria. Here's a schematic drawing of it. It's known as T-even. There's a T2, a T4, a T6. We're not real creative once again with our naming, but it kind of looks like this, almost like a spaceship guy with this little landing gears. And then this is called the head. This is the sheath, and these are the tail fibers, and it will inject the blue right here, that's the nucleic acid, and it will inject it into the host cell. So the virus, in terms of the structural components, it has that nucleic acid, either DNA or RNA, that's surrounded by a protein coat. The protein, the little subunits that make up the coat are known as capsomeres. There may be an envelope around it. Off the envelope, there may be spikes. I think everyone is familiar now with the coronavirus, the causative agent of COVID, that it has those spikes. Those spikes help the virus to attach to the host cell. They can be a wide variety of shapes. They can be polyhedral, complex shapes, helical. They may have those tail fibers like you just saw. They may have a sheath. They may be perfectly round. They may just be a rod. So they're a variety of shapes. Over here, in this picture, it is showing a virus that has some spikes on it, and another one down here, HIV, that also has more numerous spikes on it as well. And just to give you an idea of some of the helical shapes here, polyhedral, and then complex shapes. So how do we classify viruses? We use kind of a different system with them. First, just understand with viruses, we usually talk about them in families. We do not talk about genus and species with viruses. They really, part of the reason is they just haven't been studied that long to have that clear of a classification scheme with it. The way we do classify them is, number one, you look at what kind of nucleic acid it has. That's going to be like think of a flow chart, which, by the way, we call it dichotomous key. That's your first separation. Is it DNA or is it RNA? And then the next thing you look at is that nucleic acid. Is it double-stranded or is it single-stranded? So when I put something in here that says DS, that refers to double-stranded, SS refers to single-stranded. So let's say you have a double-stranded DNA, which, by the way, there can be single-stranded DNA, and there can be single-stranded RNA or double-stranded RNA. So this is where you have some of the exceptions. Most people think of DNA as always being double-stranded, RNA always being single-stranded. Viruses don't care about the rules, so they kind of do what they want. After you've determined the nucleic acid and what kind of strand it has, then you look to see, is there an envelope present or not? Yes or no. Then you're going to look at the shape. What is the host range? Remember I said it's very specific. Cell specificity. So you could say that a virus, yes, can have a human as a host. Now look at the specificity of it. Is it only going to infect, say, cells in the respiratory system, or is it going to affect cells in the digestive system? Where is it going to attack? What's its host cell? What type of enzymes may be present? Use all that to help with the classification. Now, the life cycle of a virus is a little bit different, and we use different terms for this. A virulent phage is, like I say, another word for virus, phage or phage, either one. So a virulent phage will infect the cell, and it will lead to the death of that host cell after it has replicated the virus. In other words, it's going to infect the cell. It's going to start making a whole bunch of new viruses. That's the whole point of it, is to go through viral replication and then release all those new viruses, and the host cell is going to die because it's like, I don't need you anymore because I got my job done. A temperate phage, on the other hand, is going to infect the cell, and instead of killing it, it's going to incorporate itself into the genome, into the DNA of that host cell. And if it's incorporated in there, then that means every time that cell replicates, it's replicating not only its own DNA, but also that of the phage. It may, at some later point, be induced to produce and assemble a lot of new viruses. So the lytic cycle This is when a virus is going to infect a host cell and immediately start viral replication at the expense of that host cell. Excuse me. So it's going to attach to the host cell. These are the steps of it. It has to attach to that host cell. Now, this is where the specificity comes into play. Usually there is some type of a protein receptor on the surface of the host cell that allows the virus to attach to it. If that receptor is not there, then that virus can't attach and it's just going to go on its little merry way. That is why receptor sites in different areas of the body are different, which is why the COVID virus, it's going to attach to cells in the respiratory system, usually specifically in the lungs. It will attach to some in the nasal cavity, but not on your skin. If it comes on your skin, it can't attach, so that's why it keeps going and just moves on its merry way, and there's no problem. But if the attachment sites are there, the virus will attach to that host cell. Then it's going to penetrate. It's going to, we'll see in a moment a picture, it will inject its nucleic acid into that host cell. It will immediately take over. It's kind of like in business, a company takeover. It's going to immediately take over the processing of the cell. It's going to take over the genome, the DNA, and it's kind of like, I'm in charge now. You're going to do what I need done. You're going to stop what you're doing for yourself, because I don't care. I took over this company. I took over this cell. You're going to now do what I say. And it's going to start synthesizing more nucleic acid, basically the reproduction of its DNA or RNA, depending which type it is. It's going to assemble new virus particles. So that one example of bacteriophage where it had the tail fibers and the sheaths, and it's got to start making all those new particles and then put it all together, and then when it releases it, it will lyse the cell. It will burst out of the cell. So as we've said, it's at the expense of the host cell. The host cell is going to die, and you are going to release all these new viral particles. How long does the lytic cycle take? It depends on the virus, because I mean, they all have their own unique properties. They're all a little bit different. This whole process from attachment to lysis and release of new viral particles, it may be 20 minutes. It may be 2 hours. It just depends on the virus you're talking about. How many new viral particles do you make? Once again, it's going to depend on the virus you're talking about. You might make 50, you might make 200. And what happens to all of those new viral particles? They now, in turn, go around and infect new cells. So the lytic cycle, this is a picture showing what's going to happen. You have the attachment. As you can see up here, the virus has attached to the host cell. Penetration, it is injecting, in this case, DNA directly into the cell. Most of the structure, that envelope, that protein capsule, is staying outside of the cell in this particular case. Biosynthesis, what are you doing here? You are taking over the cell and telling the cell now, I need you to start replicating the DNA. I need you to start making more tail fibers. I need you to make more the capsule heads and the sheaths. And then you're going to, so you have synthesis occurring, and then you're going to have assembly. Maturation is oftentimes referred to as the assembly stage. You're going to put all the pieces together. It's very, very regulated and organized. It's like you got to put the nucleic acid in the head, then you put the sheath on, then you put the tail fibers on. Tightly, tightly regulated. And then you're going to release lysis. You're going to have that cell burst, and all of these new viral particles go out. And like I just said, they're going to go now and infect their own cells. So the lytic phase, you're reproducing the virus at the expense of the host cell. You start that reproduction immediately. The lysogenic cycle, this is a little different now. you're still going, it's going to start off the same, where you're still going to have the attachment of the virus. It's going to penetrate into the host cell, so it's going to put its DNA or RNA in there. However, the difference now is you're going, as a virus standpoint, you're going to incorporate your DNA into the host cell DNA. You're not going to start reproduction right away. You're just going to put yourself in the host cell DNA. That's what I mean when I say incorporation of viral genome into host genome. You're putting the virus DNA into the host cell DNA. And now you're just going to kind of hang out there. This is what we call latent or latency or a dormant phase. What we call the prophase just means that is the viral DNA, that's the segment that's incorporated into that host DNA. Here's the deal. Every time that cell, that host cell, goes to replicate and go through cell division, it's going to copy that viral genome. When it's copying its own genome, it doesn't go, oh, this is mine, copy. Oh, that's not me, let's skip over it. No, it's going to copy everything. So every single time that cell now replicates, all of the new cells also have a copy of the viral genome. If you stop and think about it, that's a very effective way of just trying to kind of hide out there in the cell, let the cell copy you every time it goes to replicate itself, and you're just kind of quietly hanging back there. Now, at any point in time, you can switch from the lysogenic back to the lytic cycle. You can pull your DNA out of that host DNA, and you can pick up where you left off. You can take over, start the synthesis of the viral particles, and then assemble them, and then release them through lysis, like you would at the end of the lytic cycle. What causes you to switch from lysogenic to lytic? It may be environmental stress. It may be environmental conditions, adverse conditions, whatever, but a trigger. And you may not ever come out of it. But the lytic cycle is a way of the virus reproducing itself immediately at the expense of the host cell. Lysogenic, you're still reproducing, but not at the expense of the host cell. You're kind of hanging out there quietly. So this is showing here where excuse me, you had the penetration, attachment and penetration, the viral DNA. Here's where it incorporates itself into the host cell DNA. So like I said, every time the cell reproduces, here it is. Okay, now there's some environmental condition or stress that has triggered. Oh, okay, we need to kind of get out of here. Remove the viral DNA, take over, start synthesis of all the viral particles, assemble them together, and now release. But here's the catch. The lytic cycle, one virus, one host cell. In this lysogenic stage, if you're getting a message to switch from lysogenic to lytic, that's occurring with all the cells. How many cells have you infected if you're the virus? So greatly increased number of production of viruses in this standpoint. Transduction, just a method of transferring DNA from one cell to the other, but you're using a virus to help you do that. In generalized transduction, you're just taking a random piece of DNA and you're transferring it during the lytic cycle. You just use that virus to, it's a vehicle to move the DNA. Specialized transduction, this is when a virus is switching from that lysogenic to lytic cycle, and it may take a chunk of the bacterial DNA along with its own DNA. So here you have the virus that went into the lysogenic cycle. Now when it's getting the message, we need to revert back to lytic, cuts out. Well, notice here it's cut out a piece of the bacterial DNA as well. All that's going to be made, etc. Now, with the viruses in animal hosts, what we have found, Like I say, we look at for classification, what type of nucleic acid is there? You've got DNA versus RNA. And then you look at, is it single-stranded or double-stranded? If it is double-stranded DNA, in order to reproduce the DNA, it's going to be very similar to what you're used to. And in terms of making the protein, it's going to use the normal methods for translation. Single-stranded DNA, what happens here, it's going to make a complementary strand of the DNA so that then it temporarily becomes double-stranded. And then it can use it as normal. RNA, we have what we call plus and minus. If it's a plus single-stranded RNA, in this case, it's very, very similar to messenger RNA. So you can use it directly for translation, for the production of proteins. There are some viruses that they're known as retroviruses, like HIV, COVID is one, where what happens, they kind of have an extra step in there. Instead of using it like mRNA, they have an enzyme called reverse transcriptase. So they have this single-stranded RNA. And what they do is they use the reverse transcriptase to make a single-stranded DNA. And then from that single-stranded, they'll make another complementary so that it becomes now double-stranded DNA, and they will incorporate that into the host DNA as what we call a provirus. Some extra steps in there. Your book, don't worry, it goes into a lot of detail on all this. I'm just trying to summarize on this slide what it does. This is not a viral genetics course, which you can take. I have, but this is introductory. So I try to keep you guys from getting bogged down on some of the real nitty-gritty details of this. If it's a negative single-stranded RNA, it just makes a complementary single-stranded RNA, which would be a plus strand, and it uses that for translation. If it's a double-stranded RNA, then it just uses one of the strands to make single-stranded RNA, and it'll use that for translation. So, when we get into the section of viral diseases, I will mention at that point whether it's a single-stranded or double-stranded and what type of nucleic acid it has. But I'm not going to be certainly not picky on. Can you tell me the difference between a positive and a negative single-stranded RNA? This, as I said, is not a viral genetics course. So that's just showing how. You've got the single-stranded RNAs. They will get one of the strands to use like a messenger RNA to make the proteins. And the proteins, when I'm talking about translation and making the proteins, it's not proteins for the cell to use, it's proteins that the virus needs. Because the virus at this point doesn't really care what the host cell needs. It's just, we'll just say it's very self-centered. It's just worried about what does it need to grow. This rather complicated diagram is just showing an example for the HIV, which is a retrovirus. It's RNA, single-stranded RNA. And it just shows how when the virus attaches to the cell and then it penetrates with the nucleic acid, comes in, and it goes through this rather, I mean, really when you look at it, it's kind of like, do you have to make it really this tough? Really? Couldn't you just keep things simple? But no. So what happens is when the nucleic acid enters the cell, it's going to convert that single-stranded RNA into DNA and then make a double-stranded DNA and then finally use that to finally make the mRNA to then put back in and then finally eventually release new viral particles. Sometimes we talk about persistent viral infections, and what we're talking about here is latent infections, which are also known as dormant or chronic infections. The difference between a latent infection or a dormant infection, you've had a viral infection. Oftentimes, most cases, you've had the disease, like chickenpox. And then after you've recovered from the disease, no longer showing symptoms, the virus goes latent or it goes dormant. Basically, it hides out. Has your body gotten rid of the virus? No, it has not. The virus is still in your body. A lot of these tend to hang out in your nerves. But as the name implies, it goes dormant. And there's no signs or symptoms that you have an infection anymore. How long does it remain dormant? Well, it varies on the individual. It varies on the virus. Some of them, they go dormant and they may never have a recurrence. Oftentimes, if a virus is dormant, what triggers it to have a recurrence of the infection, sometimes it seems to be stress or environmental conditions, but other times you may have another person who's going under even more stressful conditions who also has the virus, but it doesn't come out of dormancy. So there's no way of predicting, oh, you have this virus, it's now gone dormant. Well, I can predict in 5 years it's going to come out of dormancy. There's no way of doing that. You can't. Like I say, some people it will recur, other people it will stay in a dormant state until you die. Some examples of these would be like the herpes simplex virus that causes both oral and genital herpes. The varicella zoster virus, which is responsible for causing, initially you get chickenpox. It goes dormant. When it comes out of dormancy, you end up with the rash that's known as shingles. And then the Epstein-Barr virus, which can cause several different types of diseases. Mononucleosis, casually referred to as mono, is one where it can go dormant. In a chronic infection, it doesn't go dormant. You always have the infection long term. An example would be hepatitis C virus, which causes or can cause liver damage, or the HIV, the human immunodeficiency virus. Once you have it, it's there. It's long-term infection versus just going dormant. There you go. The viral growth curve. Different stages of the growth, we give them different names. Number one, inoculation. This is when the virus binds to the host cell. The eclipse phase is also known as the penetration phase. This is when the virus then enters the host cell. Usually it's just the nucleic acid, not the entire virus with the protein code. It's usually just the nucleic acid. The burst phase, this is when the virus is going to be released from that host cell. How long does this take? Like I said earlier, it depends on the virus. Burst size. That refers to the number of viruses that are being released per cell. This also varies depending on the virus you're talking about. Typically, we usually say between 50 and 200 viruses. Viral titer. This is the number of viruses per a unit volume. And so this growth curve just shows, once again, inoculation, eclipse, the burst size, birth number. Now, this could be hours, this could be minutes. This kind of hopefully gives you a little bit of an idea. Think about this. If you have a virus that one viral particle and it infects a cell, let's say it's a flu virus, influenza virus, and it infects a cell. And in 25 minutes, that one virus that infected a cell, in 25 minutes, let's just say it releases 100 new viruses. Each one of those 100 viruses now infects new cells. In 25 minutes after that, those 100 cells now are releasing 100 new viruses per 100 cells. You start to get an idea of why maybe in the morning you were feeling perfectly fine, but after a few hours, and certainly by, say, late afternoon, you're like, you know what, I'm not necessarily feeling so good. I'm starting to feel kind of sick. but I felt fine this morning. How can this be? Well, maybe you were exposed to somebody in the morning that had the flu virus. You contracted it, and if every 25 minutes each infected cell is releasing 100 more cells, do you see how very quickly the number of viruses has greatly increased to the point where now you are showing symptoms? Maybe you have fever, maybe you have coughing, runny nose, achiness, whatever, you know, the symptoms may be. Viruses work pretty quickly. So how do you isolate viruses? Well, you have to grow them, but remember they need a host cell, so that puts another little complication on things. So you have to grow it with the host cell, and then sometimes you need to be able to separate the virus from that host cell. This is usually done by filtering. So you can grow the virus with the host cell, but when you go to filter it, keep in mind you have to be very careful of your membrane. Usually when you filter things, you do using what's known as a membrane filter. You buy these with particular pore size, the size of the holes of the filter. You can select that. This picture over here on the left shows a membrane. These little circles are the pores, and then you've got bacteria. That's what these rod shapes are. These are bacteria that are on here. So as shown over here on the right with these flasks, the two different examples, they're using two different pore sizes. The one on the left is a 5, the one on the right is a 0.2. Both of these are in microns or microliters or micrometers. You can say it either way. Most bacteria, to filter out bacteria, you usually need to have at least a 0.2, something like a 0.25 micron filter. If the size is larger than 0.2, it's going to be too big. Where, like in this diagram, it's showing these colored balls here, that the green ones, or the blue ones, excuse me, are bacteria. They're going to still be able to get through. Well, you haven't accomplished of isolating just the virus. So if you're going to filter a solution and you're trying to get just the virus, you need to be using at least a 0.2 micron filter. All the larger things, and remember, our relative idea of size is totally different in microbiology than what you're normally used to. But the larger cells are going to be held back. Only the smaller ones can pass through. This flask you would have sterilized, so it's a sterile flask. You may have had sterile fluid in it, or you may just be using fluid from here. This filter is sterile, so you're getting just the virus in it. So that's a way of getting or isolating the virus by itself. How do you grow the virus? Oftentimes in a lab situation, you're wanting to grow it so you can run tests, etc. So how do you cultivate them? You can do it either in vivo or in vitro. In vitro means in an artificial environment. In vivo means in a living organism. So you're going to have the animal or the plant hosts there. So sometimes you use tissue culture for this. You actually have tissue that you're growing that you inoculate with the virus. Sometimes a very common way is to use embryonated chicken eggs. And then if you're talking about bacteriophages that infect bacteria, you often grow the bacteria itself on what is known as soft agar. It's a little less concentrated agar. And you will grow, and on these, what we call a plaque is an area, it's a clearing spot on the surface of the petri dish where you've grown this. That clearing area is there because the virus has infected the bacteria. So we have some pictures of this. Over here on these plates, this has tissue culture. You've got cells. Oftentimes they use blood cells for growing the particular virus. Now over here on the right, under B, this is obviously petri dishes where you have probably like a nutrient agar media. You have grown when we talk about, in this case, it's saying E. coli, and you've grown, as it says, a lawn. A lawn means you have complete solid growth. That's why it looks cloudy. It's just solid E. coli growing on here. Typically, you've grown at different concentrations. The little clearing spots here, those are the plaques. You can see some here as well. Those are plaques. Those clearing areas indicate that is where the virus was present, and it was able to go through the whole lytic phase of attaching, penetrating, assembly, synthesis, assembly, and then lysing the cell. The reason it's clear is because it killed the cell. It lysed them. So that's where you know the virus is. So to determine concentration, I've had to do this before. I had the opportunity for 2 years to work with a virus, and this is the way we grew it. With bacteria. We worked with a bacteriophage, and we did exactly this. We would dilute our samples. We would have to count each one of these little plaques. That gives us a way of determining the concentration on the bacteria. We did what is known as soft agar overlays. So you had your regular media, and then you made the soft overlay that you had the bacteria and the virus in. You poured it on top of the regular media. Then you could once you had grown it and you were at this stage here, you counted them, and then you would take a sterile, it looks kind of like a scapula, and you would just scrape off that soft agar and collect that using all sterile conditions. But then that way you were able to collect the virus particles. We would have to filter it out later, so we had just the virus, not the bacteria, but we would know the concentration of it then. So that's one way of growing and cultivating the virus. Previous had the tissue cultures and the bacteria. Now, if you need to cultivate the virus, a lot of them are cultivated using, as I said, chicken eggs. I had a class where we had to do this, and I'll just say it gets a little tricky at times. We did this using the influenza virus. Chicken egg is considered a cell. You need to have an embryonated egg because you want to have the yolk sac in there for nutrients, etc. Depending on the virus determines where you're going to inoculate. Do you want to inoculate into the yolk sac? Do you want to inoculate into this lighter area right here? Is the amniotic cavity. Where do you want to inoculate? So you take your virus solution that you've prepared, and then we'll just say it does get a little tricky because, yes, you use a needle and syringe. The way we would do is we would take alcohol and wipe down the area where we were going to attempt to inoculate, and I use that word loosely because, as you can imagine, with a hard shell, it is a little difficult to try to we would just lightly tap the needle, and then once we could get it through and make our injection, we were injecting into the yolk sac when I did this, and then we'd pull the needle out. We would use tape to tape over the hole to try to prevent infection from going in. And in case you're wondering, were we successful in inoculating every egg without totally cracking and busting the egg? Nope, there were some casualties along the way. Influenza virus, a lot of viruses are grown in chicken eggs, which just kind of FYI. They use that for production of vaccines, which is why they will often ask you, like, for the flu vaccine. Flu is caused by influenza virus. That virus is grown in chicken eggs to make the vaccine. That is why they will ask you before you get some vaccines, such as the flu, are you allergic to eggs? And if you've ever wondered, why are you asking me about eggs when you want to give me a shot? It's because some of the vaccines, in order to make them, the virus is grown and cultivated in eggs. So if you're allergic to eggs, you may not be able to receive the vaccine. So that's why they ask you. One thing with tissue cultures to keep in mind when you are growing cells is that there is something that is known as contact inhibition. Most cells, when you are growing them, you start to grow them in whether it's a petri dish or a flask or wherever you're growing them, and they will start to go through reproduction, etc. When they reach the point where they are touching something on all sides, they will naturally stop growing. That's called contact inhibition. It's kind of like, don't crowd me too much. That is just something that naturally happens with most cells. It's just if they become too dense, where they're touching, at this point the growth, it's going to stop. The only way you can continue is you need to take cells out of this full plate and start again in a secondary culture. Some cells that have been inoculated with fungi, or I'm sorry, with viruses, and certainly like cancerous cells, have lost that contact inhibition nature, so that they just continue to grow and grow and grow. It doesn't matter how dense they are, and that's when you get production of a tumor. But we do see with some viruses that growing viruses in tissue culture, sometimes the cells will lose this contact inhibition property. So how do you detect if a virus is present or not? You will look at what's known as cytopathic effects. What are the effects on the host cell? Are there changes that are occurring there? You may run what we call a hemagglutination assay tests. These are typically blood tests that you're looking for certain reactions on. You may do a nucleic amplification test. This involves PCR, polymerase chain reaction, which is a process to amplify the amount of DNA so that you can run tests. You may do enzyme immunoassays. It just depends on the specific circumstances. What virus do you suspect? What are the symptoms? What disease do you suspect? That would determine how you're going to detect it. Viroids, these are short, pathogenic, single-stranded RNA. There's no protein coat surrounding it. Viroids cause diseases in plants, so it is not a problem with humans or any animals so far. They've only been found in plants. This is a type of viroid which has infected potatoes, so it's going to decrease your yield and the quality. Virusoids are pathogenic single RNA. They do infect the plants. However, in this case, the difference from the viroids is that these require a helper virus to cause the infection. They can't do it by themselves. And then lastly, the last group of your acellular pathogens are your prions. This is a fairly new area in terms of a lot of the diseases we've known about for some time, but the research is still very actively ongoing. And there's a lot of new aspects to the research into prions of what they're looking at. So what the heck is a prion? It's just, basically it's an infectious protein that has folded abnormally. So it doesn't fold the way it's supposed to, and it becomes infectious. The problem with it is that we're finding the prions are often resistant to heat, they're resistant to radiation, they're resistant to chemicals. Unfortunately, right now there is no current treatment. How do you get prions? How do you become contaminated with them? They're transmitted by eating contaminated meat is the main one. Some of the diseases associated with prions are Creutzfeldt-Jakob disease, mad cow disease, scrapie, which is in sheep and goats, and chronic wasting disease, which we are seeing in deer and elk. If you were to eat, say, that contaminated elk or deer, potentially, yes, you could develop this disease. And as I said, unfortunately, there is no cure, there is no treatment for it right now. This is sometimes referred to as spongiform wasting disease. Creutzfeldt-Jakob disease, here on the far left is showing a normal brain scan. Over here is a brain scan of someone who's been infected and is suffering from Creutzfeldt-Jakob. You notice the decrease in the density and the size of the brain tissue. And over here, when you're looking at the brain tissue, on top is the normal brain tissue, and on the bottom is one that has been infected with, it's from a patient with Creutzfeldt-Jakob disease. It's been infected with the prion. You see a lot of these open areas in this bottom one. It's often described as spongiform because it looks like a sponge. Unfortunately, you're losing a lot of the neural tissue there. It's being damaged.